Welcome to subtopic 10.1, Meiosis of higher level, topic 10, IB Biology, Genetics and Evolution. Okay, so the essential idea of this subtopic is that meiosis leads to the independent assortment of chromosomes and the unique composition of alleles in daughter cells. So when you think about our previous topic on genetics back in topic 3, we looked at that whole concept of independent assortment and how each gamete has a different assortment of chromosomes due to the processes during meiosis. So we're just going to look at that in a little more depth. Okay, so back in topic 3, we discussed there were two ways of this genetic variability. So first of all, we had crossing over, where genetic material was swapped between the chromosomes. And then we also said we had the random orientation. Okay, so the different ways the chromosomes could align before they got divided up. Okay, now, as we start looking at this whole idea of independent assortment and genetic variation, we are actually going to start seeing how Mendel's principles become disputed. And between the 10.1 video and the 10.2 video, you're going to see lots of challenges to Mendel's ideas. But if it wasn't for Mendel's initial ideas and the amount of trials he carried out, scientists wouldn't have been able to progress on to the next round of their investigations. One of the people that you are going to be looking at in this video and in 10.2 is Thomas Hunt Morgan's work, and he looked at fruit fly, and he took Mendel's work to the next level when discovering some of his fruit fly had red eyes, some had white eyes, and discovering that those fruit fly with white eyes were all males. So he started to see discrepancies in Mendel's work and looked at where that would go further. We'll be looking at your understandings and your skills together through this video. Okay, so first of all, just refresh your memory on the difference between meiosis and mitosis. Mitosis gives you two identical daughter cells, whereas meiosis gives you 4 gametes with varied genetic outcomes and only half the genetic material. So the outcome of meiosis is haploid, whereas the outcome of mitosis is diploid. The outcome of mitosis is genetically identical. The outcome of meiosis is not identical. Alrighty, so chromosomes replicate in the interphase before meiosis. So we've looked at this, and we are looking here, guys, at DNA replication. So we're thinking way back to 2.7, and we are thinking of how our DNA replicates, and that is replication. So when the chromosomes replicate, it is through this process, okay? DNA replication only has those enzymes, helicase and DNA polymerase, remember. It's semi-conservative. You always have a strand of the parent strand in the DNA that's then produced. As I said, it occurs during the interphase of meiosis. So the interphase is split into 3 phases, G1, S and G2. G1 is just your normal cell growth. Cell increases in size and volume. It moves into the S phase after it synthesizes enough ribosomes ready for cell division. Towards the end of G1, the mitochondria fuse into a network for effective energy production for the cell processes. Okay, so in the S phase, synthesis and replication of DNA occurs. During replication, the number of chromosomes remains constant a process known as semi-conservative replication. Histones are produced which wrap around the DNA copies to make them stable. This is what we call condensation. So they condense, they become smaller, fatter. You can now see them through an electron microscope. More phospholipids are produced to make the cell membrane and the membranes of the cellular organelles, because at this stage our cell is going to be going into cell division. Okay, at G2, the cell continues its growth phase as it prepares for cell division. The mitochondria divide and continue to grow. Okay, we're now ready to head into meiosis. The chromosomes have condensed and become visible as two sister chromatids. When I say visible, I mean only visible through an electron microscope. We now have the formation of our bivalents. Okay, so what are we talking about? We've got this synapsis pairing that occurs where homologous chromosomes align beside each other. Okay, this is known as a tetrad. So we've got 4 chromatids. If you look at the bottom right hand corner, you've got 2 red chromatids, 2 blue chromatids. Okay, it's bivalent because we've got 2 homologous pairs. So one is a red homologous pair, one is a blue homologous pair. You see in the picture at the top, you see you've got this filament that occurs between the 2 chromatids, that central element, okay, which keeps them aligned together. In eukaryotic cells, so this protein layer forms between the homologous pairs, and we call that the synaptomal complex. Okay, so now let's think about meiosis 1. So during meiosis 1, those chromosomes condense, okay, and the nuclear envelope breaks down and crossing over occurs in prophase 1. In metaphase 1, our pairs of homologous chromosomes move to the equator of the cell. Anaphase 1, homologous chromosomes move to the opposite poles of the cell. So they're being pulled away now. Remembering crossing overs occurred. So we've got that genetic mix-up now. And in the telophase 1 and cytokinesis, the chromosomes gather at the poles of the cells and that cytoplasm divides. So during the prophase 1, that's when this whole concept of crossing over occurs. So crossing over is the exchange of DNA material between those non-sister homologous chromatids. Okay, so it's the non-sister chromatids. So you're then swapping some genetic material from the maternal and the paternal chromosomes. So what occurs? There's a break in the DNA. The non-sister chromatid invades a homologous sequence on each other. So actually say it invades. The region of the break then binds, and these connection points are called chiasmata. It is understood that crossing over must occur for meiosis to progress. If crossing over doesn't occur, meiosis does not occur. Okay, so in your nature science, this section in this subunit is about making careful observations. Careful observations and record keeping turned up anomalous data from Mendel's law of independent assortment, and he could not account for this. And it wasn't until Thomas Hunt Morgan developed the notion of linked genes, which then accounted for these anomalies. And you'll look at linked genes in your video in 10.2. Okay, so you should have already had some idea around that. Now, Thomas More developed that notion building on Mendel's laws and the discrepancies he saw in that. You remember that Mendel thought that the outcome of crossing his F1 flowers would be the 3-to-1 ratio, and suddenly it wasn't. He had 7 other colours or something like that that he had to account for. Okay, chiasmata formation between non-sister chromatids can result in an exchange of alleles. Well, it does. That's the whole idea of crossing over. They exchange the alleles that were on that section that has been swapped. And the chiasmata is the formation of how those two sections of chromosomes join. Now, crossing over doesn't only occur once necessarily on a chromosome, it can occur at several points, okay? And you can see that here on our diagrams. Now, the chiasmata have 2 functions. One, to stabilize the bivariance, okay? And secondly, for our genetic variability. Crossing over can occur multiple times and between different chromatids in the same homologous pair. So crossing over doesn't necessarily just occur once, as I said just now. Okay, crossing over produces new combinations of alleles on the chromosomes of haploid cells. Our haploid cells being those gametes, which means the gametes have a completely new mixture of the maternal and paternal DNA within them. Okay, so thinking of what we mean by alleles, we're talking about those specific types of a gene. Okay, so you might have a gene for purple flowers, you might have one for white flowers. They are the alleles. And here we're showing crossing over, okay, down to the base level. Okay, what's happening with the bases, the base sequence. Another representation here of crossing over. Again, rather than base sequences here, we've got the alleles being shown. Okay, now one of the skills for you in this subtopic is drawing diagrams to show the chiasmata forming by crossing over. Now, when you draw these diagrams, you should show the sister chromatids still closely aligned, except at the point where crossing over has occurred and the chiasma was formed, okay? Alrighty, so we move on now and we start talking about what's going to happen there in meiosis 1. And in meiosis 1, homologous chromosomes separate. Okay, so your actual chromosomes, they pull apart. Okay, so you've got that chromosome from the father, the chromosome from the mother, and they pull apart. Okay, you can see that happening in the top row there. Then meiosis 2, it's the actual chromatid that separates. Okay, so by the anaphase here, you can see those chromosomes actually pulling apart. Independent assortment of genes is due to the random orientation of homologous pairs, homologous chromosomes in meiosis 1. And that's what we talked about. So we've looked at crossing over. Now the center picture here shows all the different arrangements of those homologous chromosomes that you could get. And because that's random, it means that the variation that occurs is also random. Okay, so we can see here in this arrangement, we can see those chromosomes, how they're going to be pulled apart, and we've got the image of it occurring there. Okay, what we've got there is the metaphase in mitosis, and you can see in mitosis, I put a big cross because I don't want you to get confused. Mitosis is just singular chromosomes aligning in the metaphase, whereas in the metaphase or in metaphase 1, it is your chromosome pairs lining up, ready to be pulled apart. Again, metaphase 1 in meiosis, and then we've got metaphase 2 there. That's showing that independent assortment as the outcome of meiosis, the different random arrangements that you can get. Independent assortment. Okay, so then our sister chromatids then separate in meiosis too. Okay, so here you can see how the chromosome, the microtubules attach to the centromere and pull that chromatid, the chromosomes apart. Those sister chromatids are now pulled apart to the poles of the nucleus. All right, so lots of key terminology during this topic, and this really just builds on what you've done in topic 3 with your genetics. Okay, so those key terms: interphase, meiosis, chromatids, the pairing, okay, that's synapsis, homologous pairs, bivalent, and your chiasmata or chiasma. Okay, so a few new terms there, but most of it is just building on what you already know. So 10.1 isn't really a unit that you should have found too challenging, because you have already covered a lot of it in topic 3. I'll see you in our next video.